Hey guys, so we are going to do vertebrate origins in this video. This is going to be the last video from this semester, and then I've got some transition videos from previous semester. So they may be a little bit older, but the content is still the same in those videos. So after this, we will start transitioning to fish and birds and mammals and amphibians in future content. But let's get started about vertebrate origins, how we start talking about cool things like us. So with vertebrates, we had all these different categories of why things are what they are. And we can see this, we've talked about these things before. We talked about symmetry first. That was one of the very first things, right? And we talked about how sponges didn't have any symmetry. And then we said the next type of symmetry besides asymmetry was radial symmetry. And that was these guys, cnidarians or jellyfish, and artenophores, comb jellies. Those are radiates. And then the next group was bilateral symmetry. Everyone else after this bilateral, just like me and you, we have a left and a right side. Bilateral symmetry. And then we divide them into coeloms. Do they have a body cavity? Do they have a true body cavity or a false one or none at all? So with our acoelomates, those are our flatworms, our platyhelminthes. Nemertea is in there. Pseudocoelomates are going to be our nematodes. And rotifers, those are pseudocoelomates. And then coelomates, you and me, that have a real body cavity. When we talk about the germ layers and the mesoderm is going to connect to that body cavity, those are coelomates, and that's basically everything else. Another category in this phylogenetic tree, protostomes and deuterostomes. We talked about those germ layers and how the cells are going to come together, or the sperm and egg come together, they fuse, they will have one cell called the zygote that makes all the other cells. This is the totipotent stem cell that makes every other cell in the body. And so that one undergoes a cleavage and it splits to all these hundreds of cells. And then that cell is a ball, and that ball invaginates in, and it undergoes why am I blanking on this? It undergoes gastrulation. That's the word I'm looking for. Undergoes gastrulation, and the germ layers are formed. It also starts to form that cavity, like our GI tract, that hole that goes through our whole body. And what opening develops first? Is it mouth first or is it mouth second? So is it a protostome? That was mouth first. Or was it a deuterostome? That was mouth second. And we can see all the protostomes. That is a huge group. All of this is protostomes. And then deuterostomes are the three guys that are left over. So we've talked about echinoderms before. Our starfish, our brittle stars, our sand dollars, all that group are echinoderms. And now we're starting to go into chordates. Before we get there, we'll talk about the hemichordates. And then we'll talk about chordates. Oh gosh, it wouldn't be a Zoe video without me yawning in them. So let's talk about what makes something transition into a chordate, and then from a chordate into a vertebrate. So there's lots of different groups. It's going to, again, include the protochordates, the first versions of chordates, as well as true straight up vertebrates. If you have the time and are curious, we have a very cool video on vertebrate origins. It is a little long. It's not one of those like 2-minute videos. It's a little longer, but it is a good video. So I've got it in here for you to talk about the history of vertebrates. Speaking of history and timelines, when we talk about eons and different time frames and different time periods, here you can see all the different groups. Here's our cephalochordates, or lancelets. When they came along, our jawless fish, and then we started getting into jawed fish. Chondrichthyes is our cartilaginous fish, and osteichthyes are our bony fish, and then we go into amphibians, reptiles, birds, mammals. All of the chordates, if you are a chordate, period, every single one of the chordates, you and me and all these other groups that I'm going to talk about, at some point have these distinguishing characteristics. They all have them. Know them, love them, memorize them. You'll need to know these for the test. Notochord. Everyone's going to have a notochord. Everyone is going to have a dorsal hollow nerve cord. Everyone will have pharyngeal slits, and everyone will have a post-anal tail. What are you talking about? What do these terms mean? A notochord. It's this guy. So we had this picture, I think we have it in 1108, our majors bio 2, but I've stuck it in here because it's really useful. We can see both of these guys in this very primitive chordate that we got going on. So here's our dorsal, here's our notochord, and the notochord gives us structure. So you and me, we have bones, right? We are vertebrates. We have a bone. We have a whole skeletal system that gives us structure. But before that was around, the notochord was around. So as we are developing in utero, we start off with a notochord, and then that notochord will devolve and change into other structures. So our notochord, specifically for us, for people, is going to devolve into our intervertebral discs in between your vertebrae that kind of give you cushion and act as joints in between the different vertebrae that allow you more mobility. That's what our notochord will degenerate into. But at first, it gives everybody structure, because not everything that is a chordate is necessarily a vertebrate. We haven't gotten there yet, but we're going to. But everybody that is a chordate has at some point a notochord. They all got them. We also have a dorsal hollow nerve cord. So your central nervous system, your brain and your spinal cord, that spinal cord is your dorsal hollow nerve cord. That's going to send all that information in and take all that information from the outside and be able to integrate that stuff and then send it out as output to the body. That dorsal hollow nerve cord, everybody that is a chordate has this as well. Pharyngeal slits are not shown. Well, they are. They are shown here, aren't they? Nice and handy. Pharyngeal slits are here. We also have a picture of them here. I like this picture best for pharyngeal slits because it gives you both approaches. We have pharyngeal slits. We did. Other organisms will kind of keep them and hold on to those pharyngeal slits because you can see they're kind of good at, here's the gill slits over here. They're kind of good when, if you were having, water flow over the gills. You know what else is good when you've got that water flowing over? If you can filter feed from that stuff. So pharyngeal slits are really useful for that ability as well, helping primitive organisms filter feed. So we had these when we were growing in utero. When we are first, like a couple weeks old, like around 6 weeks-ish, we look very much like an alien. We do not look like a person. We look weird and odd, but we have a lot of the same structures. If you look at a 6-week-old human and a baby chicken, lots of similarities there. So we look very odd. We have pharyngeal slits at this point at about 6 weeks old. And what our pharyngeal slits are, we said like the notochord will change into different structures. So did our pharyngeal slits. They are going to change into these guys, malleus, incus, and the stapes. Those are the bones in our ears. They will also change into the styloid process part of our bone and some cartilage around our larynx. It's going to have thyroid cartilage, cricoid cartilage. That is what those other pharyngeal slits will evolve into as we grow and develop. So we lose the pharyngeal slits and they turn into other structures, but we did have them at some point. So everyone that is a chordate has pharyngeal slits. Everyone also is going to have a post-anal tail. You got one, I got one. Everyone has a tailbone. For us, our tailbone is called a coccyx. My dogs are deciding to play zoomies in the background. So if you hear growling, that's what that is. They're okay. They are just being spazzes. I'm sorry in advance. Everyone's got a post-anal tail. Ours is a coccyx. My dogs have their tails going right now. But everything that is a chordate has all of these structures. They may not have them as an adult, but they had all of these structures at some point. So all of us have these. Protochordates, the very first primitive chordates before we got into fancy chordates like vertebrates, were protochordates. And there's two groups. There's urochordata and cephalochordata. There are tunicates, and there are lancelets. And tunicates only have all the structures that we talked about: pharyngeal slits, postanal tail, notochord, and nerve cord. They have those as larvae. As adults, they don't have those structures, so they look like sponges. They don't look, if you saw that, you would never think, oh, that's a chordate. No, but it did have it as a larvae, and then it develops and changes. But those are tunicates, and then these guys over here are cephalochordates or lancelets, and they're named because they look like a sword. Their lancelets look like, they look kind of like swords coming out, and they are going to filter feed with those little tentacles out there. They have a notochord that goes the whole length of their body. It's going to go down like that, and allow that notochord to act as its structure, act as its vertebrae, is what the notochord is doing for them. So muscle attachment, being able to move around, and all of that is being helped out because it's got a notochord, and it's going to have pharyngeal slits, and it's going to have a postanal tail. What's my last one? Dorsal hollow nerve cord. All of these guys, these are protochordates, are very beginnings of chordates, but they all have these structures at some point. So tunicates, urochordates, and lancelets, cephalochordates, are going to be our first versions, prototypes of chordates. Then we move on. I lied, because here's another picture of the tunicates, just to show you where those structures were as larvae versus adult, where we talk about pharyngeal slits, helping them with filter feeding and postanal tails and all that good stuff. Pictures of tunicates, pictures of lancelets, so you can see those things too. Tail, notochord, it's going to have its pharyngeal slits, and there's its nerve cord. Moving on to vertebrates. So these guys we're going to see these guys. They are going to be highly specialized, but we're going to start off with our primitive guys and then advance on there. When we learned about the different time frames that we talked about previously, we talked about, Cambrian, Precambrian, and Jurassic and all that stuff. Well, life originated in the water, right? So that's where most of this stuff is happening. We are turning from invertebrates to our protochordates to our chordates in the water. So we are starting to have, primitive endoskeletons and things like that. We think the first endoskeleton was probably going to be cartilage because it's lighter weight, and it's easy to fix and repair, and it's very, very flexible. And the cartilage here is really flexible. Some cartilage is more flexible than others. Everything has some kind of endoskeleton. If you are a vertebrate, you're going to have an endoskeleton. So again, it could be cartilage. It could be, if you are a chondrichthyes, a cartilaginous fish like sharks or rays, it's cartilage. If you are an osteichthyes, you're a bony fish. If you are a mammal, you have a bony endoskeleton that is going to give you the structure that you need in your body. It's also going to serve as a mineral reservoir. We are going to store our calcium and other minerals in your bones. And then when you need them, you can break down your bones. Literally, you'll break down your bones if your calcium levels are low, and that will help you out. So they are also mineral reserves. So that's the skeleton on the inside of your body. We talked about exoskeletons before, kind of in relation to arthropods, because their skeleton's on the outside of their body, right? The chitin on arthropods and bugs, when you crunch them, if you step on one, that's their exoskeleton. But we also have stuff that could be an exoskeleton. Your hair is protection for the outside of your body. That's an exoskeleton. Scales on fish, exoskeleton. This adorable little seal, he's going to have fur. If you're a bird, your exoskeleton is unique to you and you have feathers. All of this is a form of exoskeleton to protect the outside of the body too. Respiration needed to change. The more evolved an organism is, the more complicated it is usually, and so it's going to have systems to be able to counteract those large changes. If you are a larger organism, we said arthropods are pretty limited. They could only get so big because the cube squared law, their entire system would have to change if they got bigger. So as we got bigger, the fish needed to evolve a little bit better in their respiration. So those pharyngeal slits now evolved muscles to better increase the circulation across them, to better increase the flow. And we now have gills going on, muscular gills. If you look at a bony fish, for example, we talked about that operculum and lab, that kind of bony flap right there, that if you've ever watched a goldfish, it moves every time it takes a breath. That's all evolution, as it has to better incorporate for respiration. We are going to have a better heart. We'll see the heart evolve as we move through the vertebrates. At first, it's going to be this very basic heart that will have two chambers. The upper chambers are always atrium. The lower chambers are always ventricles. The question is, does it have one atrium or two atriums? Does it have one ventricle or two ventricles? Our primitive guys, most primitive guys are fish, and they're just going to have two. They'll have an atrium and they'll have a ventricle. So here we have our venous, our vein, blue deoxygenated blood coming in. It's going to come in on the atrium. Atriums are always on the top, by the way. Ventricles are always on the bottom. And then it's going to push it out and go to the conus arteriosus. Arteries are always going away from the heart, no matter what the blood flow is. It's always veins come back to the heart and arteries go away from the heart. So even if it's red or blue and those things are switched up, arteries and veins are always named that way. Arteries away, veins are coming in. Digestion. If you have been able to do dissections in the lab, one of the big things that you will see in our vertebrates are a big, large brown thing. Almost all the vertebrates that we dissect have this big, large, brown, blobby, loby structure. It's the liver. It's the liver. Livers are huge as far as structures and the percentage of body that it takes up. Livers are going to help us in lots of different ways. For people, livers help us detox toxins. They help us recycle red blood cells. They help us produce bile that breaks down our fats. They help us produce immunoglobulins. They help so many different ways. So liver is super important. It also is going to help, again, with bile and breaking down and digestion of food. Another thing that helps break down food is your pancreas. It's going to release a whole bunch of enzymes to help break down food. So both of these are really important digestive organs that will help and have been in our more advanced animals, in our vertebrates. Other sensory things, just like the heart evolves, the brain evolves. The more advanced the organism, typically the more advanced looking the brain is. The brain is a nerve cord. When it starts to, we said it was a dorsal hollow nerve cord, right? So it's just this tube that forms at the beginning. And then that beginning part of the tube starts to form into structures. And it will have three basic structures. It's going to have a forebrain and a midbrain and hindbrain. Forebrain, midbrain, hindbrain. That doesn't, there we go. Hindbrain. For those structures, and we won't get into the complicated parts on all those different things, but just know that it does evolve and it has these three parts, and each part has a important part, like your cerebellum and your cerebrum. All of those are different parts of the brain, and they all play an important role. I'm not going to go in there. Other sensory information, eyes. We are going to see eyes. We said that we have, instead of simple eyes, like we talked about that spiders have, we have complex eyes, and they are going to take the light in. They're going to have a lens, and they're going to reflect off of the back onto the retina. You are going to see eyes on both sides of the head for depth perception. That's going to help, whether you're a predator or prey, to be able to judge depth and see how far something is away and judge that for yourself. Another thing, these guys, you can see it right here. This is called a lateral line. We'll talk more about this in fish, but you can definitely see these on these fish, and that's a way for them to sense current. They're going to have rheoreceptors on the lateral line that helps them sense the water pressure, so they know, what's upstream, what's downstream. They can sense that stuff. Another thing that we'll talk about in fish, I see you. are going to be, oh, stop it, are going to be these guys, these little dots that look like freckles on a shark's face are really cool. And they're called the ampulla of Lorenzini. And they're all over there. Again, they look like freckles, but they're really cool because we don't have them. So I think they're extra cool. They sense electrical currents. So sharks have these, they can sense electrical currents. A long time ago, we talked about taxis. There was galvanotaxis, chemotaxis, and phototaxis. Photo was light, and chemotaxis was chemical. So we were trying to see if our planarians are going towards the light or away from the light. Do they like the food? Are they going towards the chemicals or away from the chemicals? Galvano, if you are being able to sense electrical currents, might attract you to like the motor of a boat just to check it out and see what's going on that thing. These, again, are the ampulla of Lorenzini. You can see them on a shark's face, and they detect electrical currents. So sensory information, sensory reception is getting more complicated as we evolve into vertebrates. Neural crest cells being able to differentiate into the different types. When we got all of our helper cells, our neuroglia, we are going to have cells that will branch out. We've got our cartilage and bones and our mesoderm, other connective tissue. The fact that we can branch and develop into all these different cell types is really pretty cool. Hox genes, I think I may have put it in a previous video, in a previous PowerPoint where I have a parody about what Hox genes does to Justin Bieber's song, because I like parodies and I think they're funny. But Hox genes are going to be the genes that control your body parts. They will tell when they are expressed what goes where. You know, arms attach here and legs attach down here, and my head's going to attach here. And I do not want an arm, you know, sprouting from my forehead, because that's not where it's supposed to go, because the Hox genes tell me so. It will tell the organism what goes where. You know, if I was a fly, wings go here and antenna go here and legs go down, you know, off of my thorax and abdomen. That's where the legs are going to go. And if you mess with those Hox genes, you mess with the body plan. So I want to say, I don't think it's in this class, I may have it in a different class, where they have messed with the Hox genes of poor fruit flies. And one of those fruit flies had legs coming out of its head instead of antenna because they messed with our Hox genes. So those are their body plan. Let's write it down. Body plan. What goes where are Hox genes. All right, major groups. Our first group we will talk about are fish. We're going to start with our jawless guys, which means that they just have this big sucker mouth that does not have a hinged jaw like you and I do. It's just this open, jawless type of attachment point. So we have jawless fish, then we move to jawed fish. Then from jawed fish, that's going to include our cartilaginous and our bone, our chondrichthyes and our osteichthyes. So we'll have major groups. Fish is a huge group. It is 2 lectures. Amphibians are going to be our starter group as we move out of the water and onto the land. And then we are going to have reptiles that are definitely on the land. We will, of course, see some of these guys in the water too. But reptiles do okay on land. And one of the reasons that they do okay on land is because we have evolved that reproductive egg is now an amniotic egg. So we take the water with us, and we don't have to have water like frogs do to reproduce. They don't have to. We can lay eggs, and that egg has an amnion. So reptiles are the next of evolution, moving away as far as seal, as far as animals. And then, of course, last but not least, are mammals. And we talk about the different types of mammals. We're going to have we're going to have mammals that lay eggs. We're going to have mammals that are marsupials. And we're going to have placental mammals, like you, me, are placental mammals. So those are our future groups, our future videos for the last part of this class. And those videos, again, are a little bit older, so they may be a little dated, but the content is still the same. So please, as always, let me know if you have any questions. Have a good one.